Thank you so much to the organizers for letting me share some of this work. I'm going to be talking a bit about some of the preliminary data I have coming from my PhD work, working on this animal here, which hopefully the video is working for everyone. This is Hydractinia symbiolongicarpus, and you're going to become very familiar with it through this talk. But just to give you a little quick intro, this is a colonial cnidarian. So while you're seeing many kind of individuals here, this is all one colony, all one genotype that are connected to each other. And just to give some sense of scale as well for these, this is the edge of a microscope slide. So these little hydroids right here are about 1 millimeter, sometimes a little more longer in length. So very small animals that I'm working with. Dr. Casewell already kind of reviewed some of this earlier in his talk. So I don't want to talk too much more about it other to say then my interest in venomous animals is really how venomous animals maintain or alter their venom cocktails for specific roles, either through physical partitioning, such as cone snails, or through development, as with snakes. So as they're throughout their life history, they'll change their venoms over time for certain different kind of ecological requirements. The group that I work on, though, are the cnidarians, which we just heard about. So thank you for that intro to these. These are the jellyfish, sea anemones, and corals. And though they're morphologically simple, we know that they use their venoms. This entirely venomous clade uses their venoms for a variety of ecological purposes, including prey capture, which all are carnivorous, so they do need to capture prey, as well as defense and competition. But they also partition their venoms through this decentralized venom system. So they ingest their venoms through nematocysts, which are one of the 3 types of cnidae, which are these secreted complex organelles that help define the croup. This is the rather infamous stenotele type of nematocyst from Hydra with these long barbs. And the work from cnidarians is, there's really good work on cnidarians on species like Nematostella, which certainly has changes in its venom composition over its life history. Excuse me, one moment, I apologize. So over the lifetime of the animal, it'll change its venom composition over time. There's also some really nice data on sea anemones looking at differential expression of toxins in these functionally distinct features of these animals, so specific tissues and how they're used for very specific roles. But if you're somewhat familiar with cnidarians, you might notice that all of these studies, and in fact, the vast majority of studies on cnidarians are from the anthozoans, so these sea anemones and corals. Whereas the group that I mostly focus on in the lab that I'm in works on medusozoans. So these are the jellyfish-bearing lineages. So that includes box jellyfish and true jellyfish, like we heard from before. But I'm going to be talking about a species of hydrozoan. So hydrozoans are the most speciose of the medusozoans, with a little over 3,500 species. And they're also arguably very developmentally diverse. So they're of interest to me just looking at how venoms might change over these different complex tissues and these other kind of ecological roles they might have. So this is the species I'm going to be talking about, the snail-fur hydroid Hydractinia. Hydractinia symbiolongicarpus is the North American species. I should note that Hydractinia echinata, its sister in Europe, is also pretty well studied. Both of these have been studied for several decades, and because of the community that's kind of been established working on Hydractinia, there's a lot of actually really nice monogenomic and molecular tool sets that we have, including a deeply sequenced genome, a variety of tissue-specific transcriptomes, and this species is actually pretty amenable to functional genomics approaches. It's also very easy to culture in the lab. Again, these are very small. They can spawn consistently and reach maturity fairly quickly, and they're colonial, as I said before, so you can do fairly repeatable experiments all in one genotype. But what really interested me when I got started in the lab was this feature division of labor within the colony. So like I said, all of this, this is one colony, even though there's individual zooids. The other really cool thing about this, about what you're seeing here, is that there's actually functionally distinct polyp types within this colony. So here, this zooid here, a gastrozoid, looks very much like Hydra. It has a mouth at the tip here, and will also capture prey, deposit into the mouth, and digest food for the rest of the colony. There's also gonozoids here. So this is a male colony. So these gonozoids do not have a mouth. They have a different kind of set of tentacles. And they're solely for reproduction. So in this case, sperm production, or for the females producing eggs. There's another polyp type, which I'll talk about a little bit later, that you're not seeing here, called a dactylozoid. It's exclusively a prey capture polyp. It's also non-feeding. So again, the gonozoids and dactylozoids do not have a mouth. They're not responsible for digestion. So what I'm going to talk about is kind of looking at the venom system as a whole for this animal and trying to determine if toxin composition varies across these different polyp types that have really specific ecological roles. And the first thing I want to talk about is actually something that I thought would be fairly easy to determine. It's if do these different polyp types have different stinging cells? And I say this is pretty surprising because the types of Cnidae, specifically nematocysts, and the distribution within an animal is actually taxonomically relevant in many cases. So usually within the species description of most cnidarians, you'll actually get what the types of nematocysts are and how they're distributed along throughout the life history of the animal. So I did find several studies that said Hydractinia have 2 to 3 types of stinging cells. Uroteles, which are spined and are thought to inject venom. They have an opening at the end of the thread. And desmonemes, which are hydrozoan-specific kind of stinging cell. They are adhesive and not thought to inject venom. They don't have an opening at the end of the thread, and they're very, very sticky. So they're thought just to pretty much act as a glue to catch prey over time. And the reason this was of interest to me is because later on, I'll be talking about differential expression between these different tissue types. And I really wanted to understand if I did find toxin variation, is that something about how the animals are altering their venom composition explicitly within these different zooids? Or am I really just capturing the fact that different stinging cell types have different venoms? And so that if that distribution is somewhat unique between all of these. And the way that you look at can kind of get at this question of what stinging cells are there and how they're distributed is just through looking through counting these on multiple animals. So just as to show here, so this is a gastrozoid here and a gonozoid here. If you look at the mouth, so the hypostome of this animal, you can see a bunch of these little stinging cells here. These are some of those uroteles. This is the tentacle, and you might see all these little small bunches here. These are actually desmonemes, so they have a very distinctive shape when they're not discharged. When they are discharged, they're also pretty distinctive, depending on if this thread has curled in and of itself or it's wrapped around a prey item. Surprisingly, what I saw with the gonozoids is that they do have uroteles, but they're comparatively large to the gastrozoid ones. They're actually very large. So this was already, I was seeing some sort of patterning. So that's kind of what led me to do this kind of massive counting where I took 3 different colonies, 3 polyps of each of these types from each, and counted a little over 14,000 stinging cells to get at relative proportions of each of these types. So what you're seeing is these 3 different polyp types. I counted essentially these small uroteles, large uroteles, and desmonemes throughout these different kind of parts of the polyp. So the tentacles, the mouth in the case of the gastrozoid or the feeding polyp, as well as the body column and the bottom portion of the polyp too. And so what you're seeing here are proportions of all these different stinging cell types. And there's really 2 things that struck me pretty quickly when I was looking at this the first time. One is that the gonozoid, which again is just for reproduction, so they're either making sperm or eggs, they're not participating in prey capture or digestion, have almost the same stinging cell profile in the tentacles as the dactylozoid, which is pretty much exclusively trying to capture prey and actually pass it on to the feeding polyps. So I found that pretty interesting. The other part is that when you look at the gastrozoid, the tentacle is actually almost, actually a little over 3/4 of it are these desmonemes, which again are not thought to inject venom at all. Whereas the mouth here is exclusively really these uroteles, almost no desmonemes at all. And it started to make a little bit more sense when you actually look at how these animals are feeding. So this is one of these feeding polyps, the gastrozoids, and what you're seeing is a little brine shrimp being captured and essentially glued onto the tentacle. So once the stinging cells along the tentacles have fired, this animal is not really going to go away, but it's not going to die either. There's actually been times where I've seen if these animals do escape, the animal will, the brine shrimp, I should say, will eventually get back to swimming. But what you're also seeing here is that once these feeding polyps grab onto at least one of these brine shrimp, they pretty much just shove it right onto their mouth, almost as if they're using those stinging cells along the mouth to kind of inject very directly a bunch of toxins into the animal, thus killing it, and then they're able to eat it. So this was the first thing, and this will be kind of, this will also become relevant later when looking at the toxin expression. So this next part I want to talk about is this differential expression that I did between these different tissue types. But something I want to note, and other Cnidarian researchers, and probably many of you actually encounter this issue of how the purity of the kind of sample you're looking at for RNA-seq. So one way to get around this is by dissecting the venom gland, which many of you have already talked about. Because Cnidarians don't have a venom gland, it becomes non-trivial to get a pure sample of just these stinging cells and just the stinging cell lineage to look at venom composition. And in fact, that was kind of a issue, or at least a question that kept coming up with some previous data that I've looked at. So what I'm showing here, so we actually had, like I said, really nice tissue-specific transcriptomes. And an alumni of the lab, Steve Sanders, had a really nice full tissue transcriptome for all three polyp types from wild populations. So I took this transcriptome and tried to put or annotated venom-like genes between these, and I captured about a little under 170, which is fairly reasonable for a Cnidarian. Categorized them into these biologically relevant functional groups, and then did differential expression using EBSeq. Since we had replicate data for all three of these polyp types, EBSeq allows me to essentially compare one polyp type to the other two, as opposed to doing these pairwise comparisons and then seeing what's happening. And this is the, I should say the y-axis is just the number of differential expressed genes. And it's clear that the gastrozoid has quite a bit of these toxin-like genes that are being upregulated pretty specifically to the feeding polyp. And I think what this is showing here is that really feeding is the feeding and prey capture that this particular animal is doing really requires a great diversity of these toxin-like genes. That being said, there was still this question of, are these really reflective of true toxin-like genes, or because there's not really great, because again, like I said, Cnidarians are not well annotated in terms of their, or not well studied, I should say, in terms of their venom composition, especially the medusozoans. Am I getting a lot of false positives here? So because of the great resources that we have with Hydractinia, I was able to do something pretty exciting, which is create a transgenic line for stinging cells within Hydractinia. So what I'm showing here are confocal images from this transgenic I made, which very similar to what was done in Nematostella. This red M scarlet is marking a structural protein for stinging cells called mini collagen. And what you're seeing here, so this is the feeding polyp, and this is part of the body column here, and right up here is actually the mouth. So the bit of red you're seeing here are from stinging cells that have migrated up to the mouth. And the stinging cells are develop in the body column of these feeding polyps as well. And what's kind of fascinating is that that process is happening so fast that by the time the stinging cells are fully matured, they're moving up into the tentacles, and we can still see some of this fluorescence happening here. So this is another image just of the body column. So this is where the stinging cells are being produced. And then this is the end of the tentacle. So you can see kind of this armament of stinging cells and these red stinging cells. So also similar to what was done in Nematostella, I was able to dissociate these animals and use fluorescence activated cell sorting to get a really fairly clean stinging cell sample to build a transcriptome with. And we got a really nice transcriptome from this in which I could annotate some of those toxin-like genes using the same method that I did before with the full tissue samples. And I got 105 putative toxins, so some reduction, but I would say I'm a little bit more confident about some of these because it was such a nice stinging cell specific sample. This is some of the annotation just from that. So uncharacterized and unknown are generally uncharacterized as it matched to something from a cnidarian, and had a weak similarity to something toxin-like, but I kept it as uncharacterized. Unknown is that there was no annotation at all, but I got pretty nice similarity for quite a few toxins that had been annotated, including some from box jellyfish, in fact. So, oops, excuse me. So box jellyfish toxins here, these are well-known pore-forming toxins in box jellyfish. I found some of these in this transcriptome, along with a few other box jellyfish, potentially neurotoxins in here. So I was feeling, I'm feeling pretty good about the annotation for these. And the other part that I said I was going to do with this is actually do the differential expression again between the polytypes. So now we had a really nice platform, stinging cell transcriptome platform, and I did some DE expression with EV-seq and EDGE-R for the gastrozoids and the gonozoids. And what I found was that actually only really gastrozoids really showed up as having some upregulated toxin-like genes. 51 of these 84 putative genes being what Trinity calls genes in this case, compared to the gonozoid in here. And it pretty nicely reflects actually that full tissue transcriptome that I got before of what was upregulated in gastrozoids. I think that this stinging cell transcriptome is pretty robustly capturing what is actually putative toxin-like genes within these. Why the gonozoids in this case didn't have many upregulated genes, I'm not sure. And the other thing you might notice is I don't have dactylozoids on here. And that's because Hydractinia does present a kind of interesting biological problem, and that's dactylozoids can't be grown in the lab. They can only be grown on hermit crabs. So this is the natural habitat for Hydractinia, is to actually, they live exclusively on the shells of hermit crabs. Dactylozoids exclusively grow on the aperture of the hermit crab shell. So these are some dactylozoids kind of doing this swaying motion on the edge. So we can't grow these on slides. They just don't appear on slides. So what I've done is actually take, take my transgenic line and attach it to a shell. And we've now just recently introduced hermit crabs into that shell in the hopes that we'll be able to get some dactylozoid specific tissue with this transcriptome. I know I'm nearing the end, so I just want to say that along with this dactylozoid specific data, what we're hoping to do is also use short hairpin RNA down regulation, as well as some CRISPR lines that I have working have in the lab right now to actually test the function of some of these candidate toxins that I was able to find both in the full tissue and the transcriptome specific, or the nematocyst enriched data. All right. So thank you for your time. Again, I really appreciate the chance to talk about some of this work, and I'd be happy to answer any questions.